Hello everyone, this is Harshit Jadav and today we will be talking about antibodies. Also they are known as immunoglobulins. So what are the antibodies and why they are produced inside our body? First of all, antibodies are nothing but the substance which are produced against the pathogen or we also known as antigen. So antigen के against में जो अपनी body में produce होता है, those are known as antibodies. कहाँ से produce होता है? From where? So antibodies or we can say immunoglobulins or Igs are produced from beta cells or more significantly from plasma cells. So antibodies or immunoglobulins are secreted from plasma cells. What is the basic work of antibody? So basic work of antibody is to control or to stop pathogen from invading our body as well as to initiate the immune response. So antibodies are doing offensive action as well as defensive action. How they are looking? What is the morphology of antibody? Generally they are Y-shaped in their morphology. So this is a Y shape. You can see these two flaps you are seeing, those are known as variable region, and this single line you can see, it is known as constant region. Variable region is also known as Fab region and constant region is also known as FC region. So remember this Fab region and FC region. We have also another type of morphology we can see in our antibodies. We have pentamer antibodies. So 5 this kind of antibody unit comes together and make a pentamer form. So IgM is an example of pentamer form, and the same way we have dimer antibodies. 2 same kind of unit comes together and make a dimer antibody. So IgA is the example of dimer antibodies. So the question comes, what are the different type of antibodies? So in our body there are total 5 types of antibody. Those are IgG, IgA, IgM, IgD and IgE. One by one we will take a look into all this kind of antibody as well as how they produce, in what situation they produce and what kind of action these antibody. Okay, so first we will talk about the most important immunoglobulin, which is IgG. Remember, this is the majority of immunoglobulin. It is 85% inside our body. Okay, so where IgG is found? IgG is basically found in blood, lymph and cerebrospinal fluid. So it is majorly found in blood, lymph and CSF. The molecular weight of IgG is whopping 1,50,000 Daltons. So you can imagine how big it is. Okay, so now here comes the most important part, the functions. What are the basic functions of IgG? The first important function is antibody dependent cell mediated cytotoxicity. Okay, there are so many words going on here. What is antibody dependent cell mediated cytotoxicity? So this function is basically in against of tumor. So remember, these are the normal cell and this is our tumor cell. So what happens? We already saw that we have antibody Y shape. This is a Fab region and these are the FC region. So what happens? This Fab region binds to the tumor cell. Okay, now what happens when this Fab region binds to the tumor cell? Here comes the NK cells. Now what happens when this Fab region binds to the tumor cell and NK cells comes and binds with the FC region? So here comes the NK cell. After NK cells connects to the FC region, these NK cells release cytotoxins. Okay, this NK cells release cytotoxin against this specific tumor cells. So this is the first function of IgG, which is antibody dependent cell mediated cytotoxicity. IgG defines what are the normal cell and what are the tumor cells, or against the tumor cell it will bind with the tumor cell and it will initiate the NK cell cytotoxicity response. Second is opsonization. Now what happens if hypothesize if any antigen comes inside our body? So what happens if any antigen, this is the antigen, comes inside our body? So like this IgG binds with the FC Fab region and this FC region remains alone. Okay, so this with against any antigen, IgG binds with Fab region. Now what happens? FC region will be detected by macrophages. So here macrophages will be HH. So remember, Fab region will be with the antigen and macrophage will be with the FC region. Now we all know that macrophage does the phagocytosis. So what will happen? This FC region detected by macrophages, and these macrophages will do phagocytosis. What will do? It will engulf the bacteria or the entire antigen. This process, this opsonization process, assists in phagocytosis. So this is opsonization. Third is agglutination. This is also important because when we have any antigen which is soluble in blood or lymph, then in what case, how we will be detecting or how we will be doing phagocytosis. So here IgG also plays an important role in the agglutination, because this IgG helps this soluble antigen to become insoluble antigen. Let's see how it is. So it is useful when antigen is soluble. IgG binds with the antigen, as well as in this case IgG binds with themselves. So this is antigen, this is IgG. So IgG binds with antigen, as well as IgG binds with all IgG arounds which are suspending in its atmosphere. So this IgG binds with antigen as well as binds with each other. So they will form insoluble cluster from soluble. So this way it can initiate the phagocytosis response. So agglutination helps in formation of insoluble cluster from soluble antigen. Fourth one is placental transfer. This is one of the most important, because this is the only immunoglobulin that can pass through placenta. When mother is pregnant, we all know that baby has all the immunity coming from mother's body. So how this immunity transfer from mother's body to fetus? So with the help of IgG, this is the only immunoglobulin which is helping the baby's immunity to grow. So in this case, whatever immunity mother will have, the same kind of immunity the fetus or baby will have. And the last is neutralization of toxin. We all know that we have in village we have so many snakes, spider, or a scorpion venom cases are registered. So in this case, what will happen? This IgG will bind with the active site of this venom and inactivating it. So what will happen when any person has affected with snake venom or spider venom? It does not automatically become Spider-Man. Nothing like that happens in real world. कोई Spider-Man नहीं बनता है भाई, उसको सीधा अस्पताल ले जाना पड़ता है, अदरवाइज वो Spider-Man ऊपर जाके बनता है. So what will happen? So in this case, उसको अस्पताल में ले गए, डॉक्टर इमीडिएटली गिव IgG. सो क्या होगा? IgG उस वेनम के एक्टिव साइड पे बाइंड हो जाएगा, एक्टिव साइड पे बाइंड होके उसको इनएक्टिवेट कर देगा. मतलब वी आर न्यूट्रलाइजिंग द टॉक्सिन. सो दीज आर द बेसिक 5 function of IgG. One is the antibody dependent cell mediated cytotoxicity, opsonization, agglutination, placental transfer, and neutralization of toxin. So now IgM. So as we already know, IgM has a pentamer structure. So IgM is functionally and morphologically looks like this. These are 1, 2, 3, 4, and 5 different units of IgM, and they form a pentamer structure. The molecular weight is 900,000 Dalton. It is the first antibody which was produced in human as well as expressed in human. Half-life of IgM is the 5 days. So what is function? The main function of IgM is to activate complement system. Now complement system is a major system which happens inside our body in the immunology. It is one of the most important defensive mechanism. So please learn about this complement system. Also it does the agglutination. We already see what is agglutination, same as the IgG. Now as we already saw in IgG was transferred from mother to baby, but here it is the only IgM is the only immunoglobulin which is made by fetus at the 5th month. So IgM is the first antibody which is produced in human, and IgG is the only antibody which transfer from mother to fetus. Okay, now let's talk about IgD. So the molecular weight of IgD is 180,000 Daltons. So here one of the important thing to remember is it is not secreted by B cells. It was present on the membrane of B cells or plasma cells. Okay, so and it is also used for identification of mature beta cells. If you found IgD on the presence of B cells or plasma cells, then you can know this B cell or plasma cells are mature. If you cannot find IgD on the membrane of these cells, you can say these are not mature cells. Now, what is the function of this IgD? IgD has very little function in the body's defense mechanism, but still it binds to basophils as well as mast cell and activate these cells to produce antimicrobial action in the respiratory tract. Remember, this is the only function which was identified by till date. It activates, it's bind to basophils as well as mast cell and activate them to what? Produce antimicrobial factor in the respiratory tract. It is also activate beta cells, B cells or plasma cells to activate against body's defense mechanism. So let's talk about another two immunoglobulin. First is IgE. The molecular weight of IgE is 200,000 Dalton. The half-life of IgE is 2 days. Okay, the normal half-life of IgE is 2 days. But when this IgE binds to mast cell, when this IgE binds to mast cell or the neutrophils, its half-life increases up to 2 weeks. Okay, so in the activated state, IgE is always found in the complex with mast cell or neutrophils. So what is the basic function of IgE? So first, IgE always first bind with mast cell. So IgE binds with mast cell. So this complex will bind with antigen or pathogen. When this complex comes in contact with pathogen or when they bind, they immediately release histamine. We all know what histamine is. It is one of the major inflammatory mediator. Okay, it immediately releases histamine. And now what does histamine do? What is the major function of histamine? It will give us the sign, or we can say symptom, that something is wrong inside our body. So we take antihistamine medication for that. Okay, IgG is also plays very important role in hypersensitivity reaction. This is one of the also one of the major topic comes in pathology. We have two type of type 1 as well as type 2 hypersensitivity reaction. So IgG has two basic role. First is in the hypersensitivity reaction as well as in releasing histamine. And the last immunoglobulin is IgA. The molecular weight of IgA is 165,000 Daltons. The thing we need to remember in IgA is it is only secreted in mucosal system. So inside our body, wherever you can find the mucosal system, you will find IgA. So which are the system? We have respiratory tract, we have gastrointestinal tract, we have genital tract. All this system have mucosa. So it is the only, or we can say main immunoglobulin we find in mucosal system. What is the function of IgA? IgA is only useful in we can say very several or very few diseases. First is HIV, second one is Helicobacter pylori infections. Also in streptococcus infection, there are several diseases which caused by streptococcus infection. So in this kind of bacterial or viral infection, we can find IgG. IgG has agglutination action. We all know what agglutination is. So IgG helps in agglutination to find against this kind of virus or bacteria. And Ig has very limited or what we can say normal antiviral activity. So this is the only immunoglobulin which has its own antiviral activity for the defense of the body. So this is all about antibodies. In the next video, we will talk about antigen antibody reaction, which is also one of the most important topic in the pathology to understand, or we can say concept to understand, because in the most examination this question is always there, and this also. So please like, share and subscribe this channel. Please share your views in the comment section, whether you like this kind of board activity or the normal presentation activity. It guides me well. So according to that, I will make new videos. If you like this board activity, I'll do like this. If you like presentation, I'll go that way. So this is it. Thank you very much. Have a good night.